Well, thank you for joining us today. At the end of the week, we only have a few more hours, and then we're all heading home and closing, so I appreciate you guys sticking around. I would like to describe a little bit of what we're doing at CPTx and introduce you to the company. We're an vivo CAR T company that's based in Munich, Germany, and share with you a little bit of data that we're developing towards our proof of concept and our plans of getting into an IND here in the next few years. Works. So there's two main paths that are out there right now that we've seen in terms of vivo CAR T. One approach that we've recently seen a lot of success is using non-viral vectors using mRNA, and this shows a very strong spike in protein expression, which is very applicable for the autoimmune indication space. And as some of the early indications are coming out, readouts are coming out as we speak. The second way of doing it right now at a DNA level is using viral vectors. So some of these early indications are also coming out, and the competitive landscape continues to grow in this space as well. What we're trying to demonstrate and develop is the third path to in vivo CAR T, and that's using kind of a redosable, immune-silent, and durable non-viral expression system that uses at the DNA level a LNP formulation. We want to make sure this is scalable, and we believe that this is carving out a brand new IP space that I hope can demonstrate a little bit of the early discovery data. One of the key aspects of our program is using ssDNA. So ssDNA is a very difficult modality to get a hold of in terms of large quantities and scale up. And so we've been able to develop a process to produce very large quantities that can be therapeutically relevant in a CMC process. And because of those capabilities, we've been able to develop this vector and harness some of the unique characteristics of it. One of those unique characteristics are when you use it inside of an LNP formulation and dump it into the cytosol, you get no cGAS-STING activation. As many of you may well know, when you use a plasmid DNA or a double-stranded DNA inside of LNP, and you dump it inside of the cytosol, particularly immune cells that are highly sensitive to cGAS-STING, you get a very high elevated amount of innate immune response. However, this cGAS-STING mechanism has evolved to only really detect double-stranded DNA, and as a consequence doesn't recognize single-stranded DNA, so it can in a very stealthy way go inside of the cytosol and subsequently cross into the nucleus and get secondary strand synthesis, go through as an episome. So our goal is to use an episomal vector to do transient expression. That's an alternative to mRNA and a better way than doing viral vectors. And you see we use some of the cGAS-STING markers that we always use in all of our assays to ensure that we're not activating the cGAS-STING pathway and a head-to-head with ssDNA and double-stranded DNA. This actually translates not only from in vitro but into in vivo. So in vivo, you can see using the typical panel you look for cytokine markers for innate immune response. When the head-to-head with double-stranded DNA, the double-stranded DNA has significantly elevated levels of these cytokine markers, whereas the single-stranded DNA, our vectors, show baseline results. And then subsequently, this is after a very high dose of 4 mg per kg. After 7 hours, all of these cohort of double-stranded DNA mice had to be culled, and we subsequently dosed several more times with the single-stranded DNA and saw the similar effect of just baseline results. So what this demonstrates is this vector is immune quiet and isn't getting picked up by cGAS-STING and can actually be used to deliver and edit immune cells in vivo and much more advantageous working at the DNA level without a viral vector. One of the main challenges that we had to overcome, that we kind of leveraged our capabilities in-house, is that we had to identify a transcriptionally active vector that works well. So if you look to the panel on the left here, one of the questions that we get all the time, and probably you're asking yourself as well, is what about the nuclear import? Well, we also considered that extensively on what that means for us, and what we found is that it wasn't as important as one might think. What's important is that you get a very transcriptionally active vector into the nucleus and that it persists long enough to get high levels of protein expression. And so here on the panel, you can see in the different colors, aqua and orange and red, different generations where we made over 300 different constructs over the last 8 months. We clearly demonstrated that the number of vectors inside the nucleus is the same, but the vectors definitely do not perform the same in terms of transfection efficiency and protein expression. There's a lot of know-how that went into this in terms of being able to produce all this, and we've kind of now written the playbook of how to do this. A lot of it has to do with secondary structure and the actual expression cassette, optimizing it for single-stranded DNA to specifically to T cells. The second big hurdle that we had to overcome was around delivery. And if you look into the market, obviously there are numerous mRNA LNP formulations targeting T cells. There are a few, or very minimal number, that are developed towards pDNA, and there are 0 to no LNP formulations that have ever been developed for ssDNA. So we started first with standard mRNA LNP formulations and saw absolutely nothing. We continued on to looking at DNA-based LNP formulations and continued to see nothing. So then we did an extensive panel working with lots of external partners to identify a formulation that works specifically well for ssDNA and translates over into primary T cells. And what we're able to realize is that we've identified a unique ionizable lipid that works extremely well for ssDNA, as well as optimizing the formulation ratios and a couple of the components to build a base formulation of the LNP delivery of these optimized vectors to get on par head-to-head with mRNA in the amount of transfection and protein expression. And when we translated that over into primary T cells, you can see already we're getting upwards of 30% transfection, which isn't very, I guess, a good starting point. But if you do look into the literature or in patents or other companies out there that are trying to transfect primary T cells with DNA, you'll see the best out there as a base formulation is in the 8% range. So this, as a starting point without any targeting elements, is actually a tremendous achievement that we're quite proud of. And we're already now putting on the targeting elements and conjugating CD7, CD3, and other targeting elements, and expecting to see much higher transfection efficiency over the coming weeks. The other element that we are operating on, because we're operating at the DNA level, we're able to engineer a lot of the duration and in terms of transient expression without doing genetic integration. This gives clinicians a lot of flexibility and also gives us some flexibility towards the indication that we work in. As you know, mRNA and circular RNA are very short-lived and have short half-lives in terms of transient nature. In episomal DNA, you can actually get it to last a bit longer, but because we're working at the DNA level, there's some tricks that we can play and use technology to put some genetic information in there to allow it to persist longer, either by genomic integration or doing something that we call the SMAR technology. So there's this, on the far left, you can see that this episomal persistence technology called SMARS that was developed by a gentleman that's now in our organization, Matthias Bose in Germany, and he was able to demonstrate in ex vivo in patients that you can get persistence of up to a year of transient expression episomes without any genetic integration. So what's going on here is these are very specific SMAR sequences that are engineered into the episome that associate tightly with the chromosome, which we call chromatization, which isn't integration, it's just associated tightly, and that because it's that association, it's in a very transcriptionally rich region, so you get high transcription, but more importantly, when the cells divide, they actually replicate the episome and they're carried over into the daughter cells, so you get much longer persistence. You can engineer that with synthetic SMARs, which we are currently underway doing for our lead optimization into next year, to have it last 6 months. You can have it last a year. You can even shorten it down to weeks-long persistence. And we're using this technology to keep it in the transient without doing any genetic modifications. We're also developing new single-strand DNA constructs from the market. There's many companies that are using transposase and transposon systems for integration. We're partnering with several biotech companies to help translate this modality into their system. And so we've developed a new construct around this that optimizes around the necessary activation of the transposase, where you need to have a double-stranded region. And the final area that we're looking at is to be able to transcribe right off of our vectors circular RNA, which gives us an opportunity maybe to also look at playing into the autoimmune space in the future. Should some of the mRNA companies and the products that are being developed run into some efficacy issues, we're able to actually transcribe circular RNA using some splicing technology. So our lead product that we're taking forward into next year and moving into proof of concept this year, we're planning a large in vivo study where the readouts will come in November, December this year. We've optimized the LNP formulation that I indicated earlier. We also have 3 other external partners that were optimized towards tropism that we will evaluate head-to-head. We've optimized the vector. In this case, we're going to be using CD19 for our proof of concept, but our lead product will have a dual CAR CD19 CD20, which is kind of piggybacking off of a lot of the very positive data coming back from the J&J dual CAR system. Our vector lends itself quite nicely to be able to deliver the dual CAR system in a very stoichiometric manner, which I can't go into a lot of details now. And then the last element is the targeting element, which we're doing currently, where we're introducing different targets of CD3 and CD7 on the surface with ourselves and also with a partner, as well as I mentioned earlier, some tropism-based lipid formulations with partners, and expecting those readouts in November timeframe. Our lead indication is in non-Hodgkin lymphoma, so this will be essentially we believe the first non-viral oncology transient expression system for this particular indication, which we believe separates us out from a lot of the other mRNA applications in autoimmune, and hopefully will give clinicians for the first time a lot of flexibility using the transient nature, but also being able to redose and optimize conditions for patients, giving it a much broader product profile for clinicians into the future. All of this can't be done without being able to develop the vector, and so this vector, as being the single-strand DNA, has been underrepresented, partially because it's very difficult to get a hold of. Many times there are other suppliers out there that can give you maybe a couple of micrograms to a milligram quantities, but obviously to really realize the vision of in vivo CAR T, you have to be able to deliver gram quantities to kilogram quantities. And we've been able to develop a process that uses a high cell density microbial fermentation that gives industry-leading titers up to hundreds of milligrams. And using this process that we've developed, we can get routinely or running every day 2 to 3 20-liter bioreactors where we're getting hundreds of milligrams and making those hundreds of different constructs where we basically have written the rules of how to design these constructs in terms of the cassettes and the vectors to optimize in the T cells. The other piece that we have available to us is we have a proprietary excisation technology for cutting out the backbone, where we can actually release it and open it up into a linear format and utilize some of the secondary structure characteristics that single-strand DNA can give you, further leveraging its utilization in industry for other applications. To give you an idea of what our facility in Munich, so part of it, we have the manufacturing capabilities as well as some of the early LNP discovery capabilities and immunology capabilities. We're a little over 40 people, but give you an idea, these are some of our 20-liter bioreactors that we're running every week, 2 to 3 of these. We've made year-to-date over 300 constructs, over 30 grams, it says 20 there. Our payload capacity, which we get asked a lot, is how far can I get? We've made up to 12 kb. Theoretically, it starts to tail off after a little bit after 20, but we just haven't gone that far. But we're routinely are making 10 to 12 kb. And as I mentioned earlier, we are getting a lot of pull from the market from a lot of biotech companies and larger as well to look at engineering immune cells in vivo, particularly for solid tumors, using this platform for the future. And with that, I'd like to thank you and just give some of the teams. We have Hendrik Dietz here as the CEO. He founded this company out of the Technical University of Munich and allowed us to kind of build it up. And McDavid Stillwell is our CFO, who lives in the Bay Area with me, and Stefan Oshman, who's our chairman. I'd like to thank them. Thank you.